Our last speaker of the morning is Professor Pam Ronald from UC Davis, and also she's a member of the UC Davis Genome Center and serves as Director of GRAS Genetics at JBay, the Joint Bioenergy Institute funded by the DOE. She's best known for her work on rice, the identification of genes involved in pathogen resistance and also flooding tolerance, and then taking those variants and creating new variations of rice that then had broad impact out in the world as varieties that could be used in developing countries. She's also been important in integrating researchers from around the world and establishing, among other things, the Genetic Resources Recognition Fund, which is a way to recognize the intellectual property contributions from less developed countries. Pam has received many awards. She's the 2012 Tech Award for innovative use of technology to benefit humanity. She was Fast Company magazine named her as one of the 100 most creative people in business. So you can see that she has a wide-ranging impact in plant science. And those of you who are not following plant scientists but spend time in bookstores or I guess surf on Amazon, however people find books these days, will know that she's also an author who's contributed a lot of popular articles, but also a very influential book that she co-wrote with her husband called Tomorrow's Table, Organic Farming, Genetics, and the Future of Food, which has received very good reviews. It's a very interesting book. I'd recommend it to you. So Pam is going to talk to us today about the work she's doing at JBay, looking at genes controlling cell wall saccharification in response to biotic stress.
So this project really would not have been possible without such a really fantastic group of collaborators. And there are several people in my lab that I want to highlight today. Guotian Li, who's in the audience, Ma-Sheng Chen is here, Liangrong Jiang is here, Rashmi Jen is here, Miguel Vega Sanchez is here. So please, if you have a chance, talk with them. And I'll discuss their work today. And at JGI, really this project wouldn't have been possible without Wendy and her team on the analysis and helping us set up the project. And also the Great Lakes Bioenergy Research Center. I want to give a big thanks to Nick Santoro. And Jeremy Schmutz at Hudson Alpha has really helped with the intellectual aspects on the whole genome sequencing and assembly. So as you've heard in the previous talks, really the idea is to take this large biomass, lignocellulose, and move that through a pipeline, including pretreatment, enzymatic treatment, and microbial fermentation to develop a new generation of biofuels. And one of the key issues is the recalcitrance of the cell wall. As you've heard, it's a very complex molecular components, and so one of the key aspects is to try to understand the genes that regulate cell wall biosynthesis and modification. There are several candidate biofuel crops. These are the grasses, switchgrass, miscanthus, sorghum. They're all really very exciting genetic projects going on, but we've chosen rice as a model because it is genetically very, very tractable. And it's a diploid species, has a very small genome. And you can see that it's a very small genome compared to most of the other grasses. And importantly, the genes are syngenous, and often you can take a gene from one grass, express it in another, and it maintains function. We've been working with a rice variety called Kitaki, which has really made a huge impact on our research program because it's very fast generation time. It's about 9 to 10 weeks seed to seed, and it's very easy to transform. Many of the other varieties that scientists have been using over the years, such as Nippon Bare, can take up to 4 months to generate seed, and they're also very hard to grow depending on the day length and light conditions. So we decided we wanted to develop a resource not only for our group, but hopefully in the future for other groups. So what we did, and this is work of Ma-Sheng Chen, he developed a large mutant population, and we use fast neutron irradiation. The reason we use this mutagen is because it not only creates small SNPs and indels, but also can create larger deletions, which are easier to pick out in a comparative whole genome analysis, which I'll describe today. So we call the mutagenized seed, M0. The plant that grows up is called the M1. And then our screens are done on the M2 segregating lines. So they maybe we use about 10 to 16 plants for each M1 family. And then the sequencing I'll talk about is generally done on the M3 lines, which are selected primarily to be homozygous. So the way the screen is done, they're grown in our greenhouses, they're grown to maturity, the stems are harvested, and preps, alcohol and soluble residue preps are carried out. And then we have two screens, one carried out in our lab by Ma-Sheng Chern using a hot water pretreatment, and another one carried out by Nick Santoro at the Great Lakes Bioenergy Resource Center. And then we look for alterations in sugar content and then pick the outliers and then characterize those in further generations. And this just shows you the screen. On the left is the hot water pretreatment screen, which then we evaluate for total sugar content. And on the right, we screened about 2,000 lines using the GLBRC high throughput screening. And we're primarily there looking for glucose and pentose alterations. From 4,900 lines that were screened, we identified about 40 that had an increased sugar saccharification, and 55 were a decreased sugar saccharification. I want to say these are all candidates, and we know many of the candidates fall through in the next generation. So about 95 candidates from these screens. So what do they look like? This is a slide showing you bar graphs on the left showing you the microgram sugar per mg cell wall. And there are 4 mutants here. You can see that one of them, the first one here, actually has higher sugar release in the mutant, and it's morphologically similar to the wild type mutant. The other ones have reduced sugar release, and that's more typical. We see about 2/3 perhaps with reduced sugar release and about 1/3 with increased sugar release. And then morphologically, most of the plants look fairly normal, but about 20% show a very altered phenotype. One of those mutants is shown here, which is the rice cell wall saccharification mutant 60. And you can see it's very, very dwarfed, but happily it still grows and it produces seed. This mutant has reduced sugar release, and it also, carrying out a different assay looking at the content of the hemicellulose, you can see that most of that alteration in sugar content is due to changes in glucose content. Miguel Sanchez has spent a lot of time looking at mixed-link glucan content, and so we're very interested in looking at the content in this mutant, and it's a very dramatic reduction in mixed-link glucans, and also one of the genes called CSLH1, which has been shown in barley to be important for synthesis of mixed-link glucan, is reduced in this mutant. So what are we doing now with these mutants, RCS60 and several other mutants that we've characterized in the next generation? We really are excited about the sequencing approach. So sequencing the mutant and comparing it with the wild type to identify mutations. And in the past, we use a comparative whole genome hybridization approach using a Nimble-Gen tiling array. And now we've moved to just sequencing the mutant because of the reduction in costs, and the collaboration with JGI has allowed us to use this more efficient and more thorough method. We create segregating populations. We backcross the mutant to the wild type parent, and then we analyze the F2 for segregation of a mutation that co-segregates with the mutant phenotype. So this shows you the pipeline developed by Wendy and the group in my lab, where the M3 rice is used for the sequencing, and then a DNA isolation procedure was carried out, sample submission, and then a large amount of work at JGI for quality control and library prep, and then Illumina sequencing. So this shows you an example of some of the reads where you can identify a SNP, and this is a to G mutation that you can see. So on the top is the wild type genome, and then the sequence that are obtained are shown, the reads are shown below. And you can also identify small 10 base pair mutations. This is a small deletion. And then on the right, we can confirm that by PCR. So you can see the wild type compared to the mutant, where the mutant has a small deletion that can be detected by PCR. So this verification is very important for this project. So where are we now? So our goal is to create a sequence rice mutant collection of 2,000 genomes. To date, we have JGI has produced 178 rice mutants, and we're actually very excited about this because it's the prep that takes a lot of time to develop the procedures that work for both groups. JGI is predicting that 1,000 mutants will be sequenced this year, and we're aiming for a total of 2,000 by 2016. We then need to identify genes corresponding to mutations. So we take those candidate mutants that we've developed so far, we're making back crosses, and then we'll be able to look at co-segregation. We've also begun to characterize the frequency and nature of mutations in our populations, and this is also quite exciting. So I should mention mutagenesis has been used for a very long time, nearly 100 years, for genetic analysis in plants and animals, and it's also been important for breeding approaches, because when you characterize, you develop induced mutant populations, then breeders can select those lines that carry traits of interest. So mutant populations have been around and have been very useful for geneticists and breeders, but often we don't really know how many mutations per line. And so this allows us to really quantify the number of mutations and the nature of mutations. We've just begun this analysis, but this shows you in analysis of the first 45 lines, the frequency of the mutations. And this slide only shows the SNPs, or small indels. And just looking at this calculation, you could see about 70% are insertions or deletions, which is predicted by fast neutron populations, and about 30% SNPs. So, and these are SNPs or indels in coding regions. So there's about an average of 7 SNPs or indels per gene in a mutant line. So, but this only captures part of the mutations. There's also larger deletions and insertions that aren't captured yet in this analysis, and we're working on that. But we predict that each line will have about 15 to 20 knockouts in different genes. So we're also then able to begin mapping these, and this shows you the 12 rice chromosomes, and you can also see the mapping. So we have insertions, deletions, and SNPs are shown here in 3 different colors. And you can see that the mutations are fairly well spaced over the genomes. And so this will be important as we develop more and more of the mutations. We can map them all, which will be useful for other researchers. So so that's where we are with the cell wall saccharification screens, and I should mention that there have been many other mutant populations developed in rice, and they've been useful to our lab and other labs. However, most of those mutant populations are made, all of those other mutant populations are made in different varieties, such as Nipponbare or Korean varieties that are often more difficult to work with and propagate. And the access to the collections are not always easy. There are international shipping regulations, and so sometimes it's actually difficult to get those lines. So we're hoping this population will be useful to many researchers around the world. I wanted to also talk about another project in the lab, which is resistance mediated by the XA21 pattern recognition receptor. This has been an important gene that we studied in the lab for a number of years, and we're also using the mutant screens on this variety as well. So it's the same mutant population, but we're screening for alterations in resistance rather than alterations in cell wall saccharification. So this slide shows you it's the bacterium on the left. It's a gram-negative bacterium, produces a yellow pigment. You can see that when the rice leaf is infected, it oozes out of the plant. You can see the bacteria coming out and also the water-soaked lesions. And this disease causes a very serious disease in southern Asia and in parts of Africa. So a number of years ago, we isolated the XA21 gene for resistance, and you can see here on the left are the parental variety, in this case called type A309, and we take scissors, we dip them into a bacterial suspension, we clip the top of the leaves, and we measure lesion length analysis. On the right is the transgenic line carrying the XA21 gene, and you can see those lines are very resistant to bacterial infection. This XA21 encodes a receptor kinase, a classical receptor kinase with leucine-rich repeats on the extracellular domain, a transmembrane domain, and an intracellular kinase domain. This structure gives us an idea for how XA21 functions. We hypothesize that the extracellular leucine-rich repeat domain recognizes a microbial molecule and that recognition triggers a downstream defense response. XA21 has remarkable structural and sequence similarities with other important proteins, such as the toll receptor of the toll receptor of Drosophila, which you can see also has a leucine-rich repeat domain, a toll interleukin receptor domain, and this receptor associates with another kinase domain called Pele. This toll receptor is critical for resistance to infection, microbial infection in flies. There is another large class of plant resistance gene called NBS receptors that share similar domains with the fly toll receptor and the XA21 receptor. And in mammals, the TLR4 receptor is critical for recognition of pathogens and critical for the innate immune response, which is a very important discovery in animals. We now know that other plant species, this is Arabidopsis FLS2, share a large number of these types of receptors and another human receptor called TLR5. So many researchers in the fields are now trying to understand the downstream signaling approach, and we've used a number of molecular techniques, and today I wanted to mention the work through the mutant screen. So Masheng identified 3 mutants that were altered in the XA21-mediated immune response. So for this screen, he grew these in the greenhouse, transferred them to a growth chamber, and inoculated, and simply looked for mutants that were susceptible to the pathogen. So this shows you the 3 mutants. On the left of each of these panels is the XA21 line, the parental line, which is highly resistant, and then 2 leaves from each of the mutant lines, and you could see they're highly susceptible to the pathogen. And this is an enormously challenging screen because it takes a lot of greenhouse space and a lot of patience. But happily, the work has been going very well, especially with the collaboration with JGI. On this mutant, FAS neutron 1916, there are three deletions, one on chromosome 2, one on chromosome 3, and one on chromosome 4. And once those deletions are identified, we can develop primers and then look to see if that mutation co-segregates with the mutant phenotype. And this shows you one example. On the left, then, in green, you can see the wild type has very short lesions, and then the control line in blue has very long lesions, and then the progeny, the F2 segregating progeny, are then shown to the right. And you can see that every time we see the wild type band, the lesion length is low, and every time there is a deletion of that band, the lesion length is very high. So this indicates that there's a co-segregation of the mutant band with the mutant phenotype. And similar results is in FAS neutron 2405. So far we've identified one deletion, and this deletion co-segregates with susceptibility in this mutant. So the next step is to look at these regions, and in FN 1916 there are 2 small uncharacterized genes, and in FN 2405 there's about 15 non-transposable element genes. So for FN 1916, since there's only 2 genes, we're planning to we've begun to complement the mutant with each candidate gene to see which one of these genes complements the mutant phenotype. And for the larger deletion with carrying 15 genes, we decided to silence each candidate gene and generate transgenic plants, and then complement the mutant with the candidate gene. So this mutant population can also be used for reverse genetics analysis, the sequence with the help of the developed sequences. So I wanted to briefly mention a project that was initiated several years ago with Edward Marcotte at the University of Texas, Austin, who is an excellent computational biologist, and In-Suk Lee, who's now at Yonsei University. They developed RiceNet, which is a network that was developed as guilt by association analysis. They used 50 million data points and 23 types of data sets, and from 5 different species. And this network analysis is very useful because instead of only looking at a single data set, for example, a rice transcriptional data set or a protein-protein interaction data set, RiceNet then gathers all that data together, as well as data from other plant and animal species, which we know have very similar signaling pathways. And you get this beautiful piece of art, which is actually Edward's group presented this Arabidopsis network at the Museum of Modern Art in Austin, Texas. So it's art and science at the same time. And we've used this then to query this network. So what we do is we're able to take XA21 and then other genes that we've shown to be critical for the XA21 mediated response and query the network. So the red balls are XA21 and the other proteins, and then by querying the network, we can identify, in this case, hundreds of other genes shown in green that are predicted to be involved in the same biological process. Now, this work leads to hundreds of genes. It depends how you do the analysis, but this particular analysis led to about 800 genes. It's way too many to validate in vivo using your classical transgenic overexpression silencing approach. We did do that with about 5 of these genes, and we're able to validate that the network is very useful and does highly predict genes, but it takes a long time to do the transformation. So that's one reason we're very excited about the sequence collection, because now we can do reverse genetics. We can then go into do these different types of queries and then immediately go to the collection and have the seed packet that carries a mutation in the gene of interest. And this is important, as I mentioned, not only for analysis in rice, but for example, if there's genes that people are looking at in other type of grass species, they can then quickly do the analysis in rice. So we have 7,000 Kitaki mutant families. The goal is to sequence 2,000 of these in the next couple of years. And then Rashmi Jain, who's here in the audience, is developing Kitbase, which is a database of the Kitaki mutants that will be a really important tool for reverse genetics. So it will carry location of the seeds, as well as the phenotypes and the genes. The phenotypic information that we will start with in the database are cell wall saccharification, disease resistance, developmental phenotypes, grain size. And we hope to eventually expand the other phenotypes in this database, and hopefully someday we'll be able to bulk up enough seed that we can distribute the seeds to other groups that are interested in doing mutant screens and identifying genes corresponding to the mutations. So, to summarize, we have screened about 1,500 fast neutron mutant families in the cell wall saccharification screen. We've identified 95 candidates that are now being confirmed in the M3 generation. Those that are confirmed will carry out back crosses and do genomic sequencing. The ultra-resistance screen is essentially complete, and we've identified 10 mutants that have been confirmed, and already 2 of them have been shown to carry deletions that co-segregate with the mutant phenotype. So we really are excited about this approach and believe that whole genome sequencing greatly facilitates identification of genes corresponding to mutations and will be a very useful research tool to the GRAS genetics community. And we hope to have Kitbase available in about 2 to 3 years. So thanks again for the many people that collaborated, and thank all of you for joining the talk today.
Thank you very much, Pam. We have time for a few questions.
Hi, Pam. I just want to ask, when you have sequenced 2,000 genes, can you estimate, based on what you know now, how big a fraction of the total open reading frames that you would have targeted in that population?
We were just talking yesterday about doing that calculation again. We can estimate that, but we haven't yet. So first we need to get a better number of how many gene knockouts per line. And we should have that information available probably in the next few weeks based on 45. So if 45 is representative of all the 2,000, then we'll have that number, and then we can figure out how many. I think 2,000 is going to not be at 90% probability of identifying a gene in your interest, but it might be up at 70 or 80%. We think we need to probably go to about 4,000 if we're going to have a 95% certainty of identifying a mutation in a particular gene of interest doing reverse genetics.
You have 7,000, just so keep sequencing. Or...
Tell our collaborators here.
You said that you had about six lesions in coding sequences per line. Is that about right? That was you had a distribution.
Yeah, on average about 6, but those are just the SNPs and the very small indels less than 30 base pairs. So we actually know there's much larger ones, but it's taking a little bit more time to get that information.
Right, and not all of those will have loss of function, so that affects this calculation too.
Yeah, that's true. Although the loss of function, I think we have to calculate it that if there's a mutation in a gene, we have to just assume it's a mutation that will cause some alteration in function in order to make the calculation. But you're right, the SNPs maybe, even if you change the coding, perhaps it'll still have function.
Right. Is there a question over here? Sam.
I wanted to ask you about the non-coding mutations. Have you ever found examples of non-coding mutations that produce phenotypes? Do you look at those?
We don't have that data. So Guotian's sitting next to you. He can answer the question. I mean, you would see the mutations in the non-coding regions, but they're not quantified, right?
Yeah, we are still in the process of analyzing all this data. You know, you see in Pam's talk, for a particular mutant, we did that by manually, so we see the big deletion, but for the whole mutant population, we're still working on. But definitely, from our data, there are much more mutation, no matter SNPs or indels, in the non-coding region. That's, I think, the predictable.
Any other questions for Pam? Any other questions? If not, I want to thank all of our speakers. It's been a wonderful session this morning. We have a lunch break, and then we'll come back for the technology session, which will include the traditional discussion of the new technologies for JGI and what the community needs. So thanks again for Pam and all the other speakers.